Okay, let me get it. My name is Jason, and I'm in the University of Virginia. Today it's my great pleasure to introduce my friend and colleagues, Dr. John Luken. So a little bit background of John. He did his bachelor degree in University of Richmond in Virginia, and then received his PhD at the UVA University of Virginia with Dr. Yang Hang, where I get to know him when I was a postdoc with Tom Bracciali. After receiving his PhD, he went on to Tiro Kaliganti's lab, who just gave a seminar a few weeks ago. So John was in Tiro's lab for like 5 years, really set like a sky high standard for in terms of productivity. He published 5 first author papers in Tiro's lab, 2 in Nature, 1 in Immunity, 1 in PNS, and 1 in GI. So very, very productive during his postdoc. So in 2014, he returned to UVA. Since then, he continued to be very productive with his own lab and published several very well-regarded manuscripts, including a paper in Nature, which is also going to be his topic today, on DNA damage, innate inflammation, and neurodevelopment. So John is supposed to say is a rising star in neuroimmunology. So he received a few awards, including the Hardware Foundation Investigator and the Siemens Foundation Autism Research Investigator. I have been known John for like 15 years. So for just my own one sentence to summarize my what I see him, he's very smart and sharp, like a razor, but at the same time very considerate to other people, very engaging and extremely funny. He's very well regarded and extremely popular at the UVA. He also gives great speeches, and so I hope you all will enjoy his talk today, just like me. So without further ado, John, stage is yours.
Thanks, G. That was quite a nice introduction. I wish you could give all my introductions. Yeah, I've known G for 15 years when I was a little scared graduate student, and G was a rising star working on IL-10 in Tom Bracciali's lab. Super happy to have him back as a friend and a colleague. So we'll get started. I don't know how you guys deal with questions, but I'm happy to stop if there's any major questions, and also obviously answer some questions at the end too. So most important slide first. So all this work was done by Dr. Catherine Lammert. So Kat was the first graduate student to join my lab when I started in 2014, and she did an incredible job. She's now at Northwestern doing a postdoc studying autism. But all the work that you're going to see today was done by her. And it's just amazing what she was able to accomplish as a graduate student. So a couple of years ago, we became interested in whether the innate immune system was being activated in neurodevelopment. And then obviously, if it was this contributing to the shaping of the developing brain? And the reason we were curious to know if there was any engagement of the immune system in neurodevelopment was that it's known that during this period, there's massive cellular proliferation in a short period of time. And sorry, I'm just checking the okay. I was just checking this chat. I should warn everybody, I am at home and I have a dog that's sometimes very poorly behaved. And G and I also, there was a tornado warning. So if you see me up in the sky at some point, the tornado picked up my house. So back to the talk. So we were interested to know whether the innate immune system was shaping neurodevelopment. And this was primarily because we knew that during this period, this was a time when there was lots of potential triggers that could engage the immune system that were being generated. So you have this massive neurodegeneration or neurogenesis, so you proliferation of neurons, and they're differentiating, and this causes a lot of metabolic stress, also a lot of DNA damage. And then what happens is the brain produces over half of the actual neurons and other cells that are produced in the developing brain actually undergo dieback. And this has to be properly gauged. And if you get not enough dieback or too much dieback, you have neurodevelopmental disorders. So during this process of neurodevelopment, there's quite a few triggers that could engage the immune system. And we were curious to know, was this occurring? And then what are the implications of that? So we turned our attention to the inflammasome, which is probably one, if not the most important immune sensor for various forms of stress, both stress associated with pathogens and then also stress that occurs endogenously, things like mitochondrial perturbations or other types of endogenously derived stress. And just briefly here, I'm sure this is well known within this crowd, but for those that are maybe outside the field of immunology that are tuning in, what inflammasome is a multi-protein complex that forms and includes an inflammasome receptor such as NLRP3 or AIM2, an adapter protein, which is called ASC, and then caspase-1. And it's this formation of this multi-protein complex that puts caspase-1 into the right orientation. And once it's in this orientation, it cleaves itself, it becomes activated, and then it can do a bunch of things downstream. So it can cleave and activate two very pro-inflammatory cytokines. Those are IL-1 beta and IL-18. And it can also cleave and activate a molecule that you're going to hear more about later, Gasdermin-D, which forms pores in a cell. And if you get enough of those pores that are formed, it can generate this lytic force to cause cell death. Importantly, many of the actual triggers that we know are present in the developing brain, including damaged mitochondria, DNA damage, changes in potassium flux, dead cells, changes in cellular volume, and ER stress are known to engage this pathway. This is well known in looking at macrophage and classical immune cells, but we were interested to know if it was also potentially being triggered in the developing brain. So I've already stated this. We wanted to know if inflammasome activation was occurring in neurodevelopment in response to these potential triggers, which we know are present. And we were curious to know how this shapes the developing brain. So the way we did this was we used an ASC reporter mouse. And this was generated by Doug Golenbach a couple of years ago. And it builds off the premise that when inflammasome activation occurs, you have ASC forming a puncta that forms this multi-protein complex. So all the ASC that's usually dispersed throughout this cell in the cytoplasm will converge usually into 1-2 puncta per cell. And this is called an ASC speck. And this is commonly used as a readout in inflammasome activation. So this is from Doug's group. It was published in Cell Reports in 2016. But what they showed was typically ASC under resting conditions is diffuse in the cell. But if you engage it with known activators of inflammasomes, you can see that ASC specks form, and this is used as a readout for inflammasome activation. So we looked in the developing brain using these ASC reporter mice, and we looked at P5. And we chose P5 because this is a time point where you have massive dieback of neurons and other progenitor cells in the developing brain. We also know that there's lots of reactive oxygen, metabolic stress, and also DNA damage at this time point. And so this is P5. So this is 5 days after birth, which in humans correlates closely to the 3rd trimester. And we were surprised to find that in various places throughout the developing brain, so this is at P5, there was quite a bit of speck formation present, and this was much higher than we were predicting. So this is looking in the cerebellum, which is involved in motor coordination. We see in the developing brain a little bit of speck formation in the hippocampus. We see some in the cortex. Virtually every place you look, you can see it, and it varies depending on when you look, where there's enrichment of speck formation. And if you compare this to a classical immune organ like a lymph node, so this is a resting lymph node, so there's no activation at all. This is homeostasis. You're lucky to find one speck in a resting lymph node per slice. And so this is just as quantified. Very few, if any, specks are found in the resting lymph node. But if you look at the developing brain during development, you see that there's quite appreciable levels of inflammasome activation, much higher than we were predicting. And we validated this using the antibody to look at endogenous levels that aren't associated with the fluorochrome. Okay, so this suggests that yes, there are some triggers present in the developing brain that can activate the inflammasome. So is this just a bystander effect that really means nothing, or is there potentially a biological effect of this inflammasome activation in terms of shaping the brain? So the way we did this was kind of a reductionist approach. We first ran a battery of different behavioral tests to evaluate if the neurological function was altered in mice that lack inflammasomes. And this is just to get a broad idea of, if you don't have inflammasome activation during development, would there be an impact on classical neurological function? So one of the first things we looked at was the elevated plus maze. And for those who haven't run this test before, the premise is that even though we raise our experimental mice in captivity in our vivariums and our mouse rooms, they still have this primal fear of potential predators like birds. So if a mouse is, the thought is, if a mouse is anxious, it will not go out into open spaces, and it'll retreat into closed spaces, which are protected from these potential predators. So this maze builds off of this theory. It's a plus maze. So 2 of the arms are closed, and then the other ones are open. And this is a typical wild-type mouse. And you can see the O refers to the open arms. And you can see most of the time is spent in the closed arm, but this mouse, like many other wild-type, will go out into open arms and see what's going on. In comparison, mice that lack the critical inflammasome effector molecules, that is caspase 1 and 2, or caspase 1 and 11, my apologies, also known as ice knockout mice. So these are mice that are deficient both in caspase 1 and caspase 11. You can see that these mice are very anxious, so they very rarely go out into this open arm of this test. And this is combined data showing here that they spend much less time in the open arm. We've also validated this by another anxiety-related behavior test. This is known as the open field test. Same premise as the elevated plus maze, where mice that are anxious will not go out into open areas. So this is just basically a box, a plexiglass box, and you just track how the mouse spends its time in this box. And you can see here, wild-type mouse is making many trips out into the center to explore, whereas mice that lack both caspase 1 and caspase 11, which are the critical effectors for inflammasome activation, those mice are spending much of their time in the corners and very rarely entering out into the center, suggesting that they're presenting with somewhat of an anxiety-related behavior. So you can see this is combined data. So inflammasome-deficient mice have less bouts into the center, and then they also spend less time in the center. Another major neurological feature that we see in these mice is that they have incredibly bad motor coordination control. So the way that we test this is using a rotarod. This is like one of those log roll tests that you see at some carnivals and stuff, where you can see how long you can stay on the log in the pond or the lake. So basically, the rod accelerates, and if you have issues with motor coordination, you fall off sooner. And you can see with mice lacking caspase 1 and 11, so the critical inflammasome effectors, that these mice have very poor motor coordination. So these mice aren't showing, so it appears that most of these neurological impairments are quite specific to a number of behaviors. And it's not just that these mice that lack this critical immune molecule are showing some kind of sickness behavior or potentially have an outgrowth of some kind of infection in the mouse strain. Because when we look at a number of other tests, so this is a locomotor activity test where they can see the platform in a pool, and you can see that they're very active and can find it just in similar times as the wild type. We've also looked at depressive behaviors. So this is a sucrose preference test, and they're very similar to wild types. In the tail suspension test, which is another test to use to evaluate depression, we see that there's very little, if no, impairment. I'm not going to show the data here today, but we actually see that some of these mice are actually smarter in some of our behavioral tests too. But that's a story for another day. So having seen that there was inflammasome activation in developing brain, and that if you don't have this inflammasome being engaged during their development, that there was impairments, we next wanted to get a better sense of what the inflammasome was responding to, and in hopes that that would give us a better insight into why this was occurring and how it could potentially help to shape a healthy brain. So this crowd probably doesn't need this introduction, but there are obviously multiple sensors that can generate inflammasome activation. And you could probably, there's additional ones that you could add here, NLRP6 being one of them. But we turned our attention primarily to those sensors that respond to triggers that would be present in a sterile environment. So things that are endogenously derived danger or damage signals. So what I'm talking about there is NLRP3, which is probably the most famous and well-known of the inflammasome sensors. And it's very promiscuous and can respond to various different triggers, things like metabolic distress, reactive oxygen species, necrotic cells, various different stress signals that could be present in the developing brain. Another potential trigger that could be contributing to the inflammasome activation in neurodevelopment is AIM2. So AIM2, in addition to responding to double-stranded DNA that's pathogen-derived, can also respond to endogenously derived genomic DNA damage that gets out into the cytosol. So we turned our attention to these two sensors to see if they were involved in shaping neurodevelopment, and then obviously the behavioral functions. So what we noticed, or what we found, was when you knock out, and these are all germline, so full body knockouts, when you knock out NLRP3, these mice don't show any anxiety-related behaviors, so they're going out into the open arms just as much as what we see in the wild type. And this was somewhat of a surprise, because we were going into this expecting that a molecule like NLRP3, which could respond to so many different triggers that are present in the developing brain, would be the major culprit here. But instead, what we found was when you knocked out AIM2 and full body knockout, that these recapitulate what we were seeing in mice that lack caspase 1 and 11, suggesting that AIM2 was likely the upstream sensor that was engaging this biological pathway. And this is just a combined data here showing that there's really no effect on anxiety-related behaviors with NLRP3 deletion, but we do see something similar to what we were seeing in our inflammasome knockouts with AIM2 deficiency. We see the same thing in many of our other behavior tests. So NLRP3s are fine in this open field test and going out into the middle, whereas the AIM2s look a lot like the caspase 1 knockout mice, spending very, very little time in the center. And as before, this is just our combined data showing that we get a nice phenocopy of these behavioral abnormalities between the caspase-111 knockouts and those that lack AIM2. So earlier I showed you speck formation in developing brain at P5. And we next wanted, now that we saw that AIM2 was a major contributor to some of the behaviors, we want to see if that sensor was also involved in driving the inflammasome activation that we see in the developing brain. So what we did is we crossed our AIM2 knockouts onto the speck reporter mice and looked at this. And what we found was that when you do knock out AIM2, so this is just a DNA damage sensor that can activate the inflammasome, you see that this knocks down quite a bit of the speck formation that we were seeing in the developing brain. So suggesting that, yes, this AIM2 DNA damage sensor can coordinate much of the inflammasome activation that we're seeing in the developing brain. So earlier in the introduction, I introduced broadly what the inflammasome was, a multi-protein complex that activates caspase-1. So downstream of it, many things could happen, and we wanted to get a better sense of whether this phenotype was being driven by a deficiency in, say, IL-1 or IL-18, which, in case you didn't know, and this is something I didn't know until I started my lab, IL-1 and IL-18 can impact neurodevelopment in numerous ways. But that's a story for a different talk. Or also, could it be through impaired cellular dieback downstream of DNA damage? So to do this, we looked at mice that were deficient in the IL-1 receptor or the IL-18 receptor. And what we found was when you knock out both of these receptors, it really has no impact on the behavior or any appreciable effects on the speck formation. We've also looked in a MyD88 knockout, so downstream of the IL-1 receptor, and then also the IL-18 receptor is MyD88. So this would control for a compensation between IL-1 receptor and IL-18 receptor signaling. Next, we looked at the Gasdermin-D knockouts. So as before, these are full body knockouts. And as I mentioned in the introduction, Gasdermin-D gets cleaved by caspase-1 and can form these pores. And if you get enough of those pores forming, you have this lytic form of cell death known as pyroptosis, which is Gasdermin-D and triggered by these pores. So what we found looking at our behavioral assays was that, like the caspase-111 and the AIM2 knockouts, the Gasdermin-Ds phenocopy the behavioral abnormalities. So this is the elevated plus MAGE that we use for anxiety-related behaviors. And you can see that there's a very nice consistency between the behavioral defects in this anxiety-related behavior test. We also see it in various other of our behavior tests, including this rotarod, which looks at motor coordination. So having seen that there's a DNA damage sensor that can trigger inflammasome activation, and this was required for normal neurodevelopment, and if it didn't happen, you would have behavior abnormalities. We wanted to get a better idea of why this was happening, what kind of beneficial effects it could have. And one of the ideas we came up with was perhaps AIM2 and the inflammasome was engaged as kind of a backstop to remove or purge the brain of any genetically compromised cells that were being developed during development. And the reason why this is especially important in the developing brain is because many of our CNS cells are extremely long-lived. So neurons obviously live hopefully for the duration of our lives. If they don't, we tend to get things like neurodegenerative diseases as we age. But even glia, things like microglia and astrocytes, which make up the brain and also play very important roles in neurobiology, they're extremely long-lived too, in comparison to, say, like a peripheral macrophages that's in circulation. So we're talking about years and years and years. So obviously there is this added pressure to get things right straight out of the gate and to limit any kind of wiring of cells into the developing brain that would have DNA damage. So this was obviously to be completely honest, we never really planned to pursue this experiment in this detail. But once we got down the road, we thought this was a pretty interesting area because there's quite a bit of data looking at how DNA damage is regulated in the developing brain. And much of this work was done by a whole group of people, but Peter McKinnon has some amazing review articles on this topic. But as I mentioned in the introduction, there's various points in neurodevelopment where DNA damage can be accrued. So obviously during neurodegeneration or neurogenesis, where you have these proliferating cells that are just going gangbusters, you can have a lot of replication stress. As they differentiate, you're looking at a lot of oxidative DNA damage. And then once they've actually matured and they're starting up their circuitry, you could still have oxidative damage, but you can also have transcriptional disruptions that lead to DNA damage. And there's various different forms of DNA damage that can develop in the brain that's undergoing maturation. And this has led to this prevailing paradigm that maintenance of genomic integrity is a prerequisite for CNS health, in that if you can't control or properly handle DNA damage that accrues during development, that this could have pretty dire consequences on the health of the brain. And these are just some examples of some neurological disorders that are associated with DNA damage repair proteins. So basically every DNA damage repair protein that you can think of, although is probably most well known for causing things like cancer later in life, many of these loss of function mutations also lead to early onset neurological diseases. So probably the most famous one is ATM, which when mutated can cause ataxia. And many of them, the other ones, can cause some early onset neurological disorders. This is a pretty severe one where you have really aggressive aging. So this is all well and good, nice theory. We wanted to first understand, okay, if this is the case, we wanted to see if the AIM2 was actually coordinating cell death in response to DNA damage. So we first took an in vitro approach where we took bulk neural cells. So this includes both neurons and also glia, like microglia and astrocytes. We've also done it with just neurons. And we induced DNA damage. And what we're doing is we're comparing those to mice or to cells that lack either AIM2, the DNA damage sensor, caspase 1 and 11, the effectors of inflammasome, and then Gasdermin-D, this coordinator of cell death downstream of inflammasome activation. And one of the first things we did was looked at ionizing radiation. So this is known to be a major trigger of DNA damage and cell death. And you can see that the wild type cells are undergoing quite a bit of cell death in response to ionizing radiation induced DNA damage. However, when you get rid of AIM2, caspase 111, and Gasdermin-D, you can see that this blunts the cell death that's occurring, suggesting that the AIM2 inflammasome and Gasdermin-D are at least one pathway that can purge or remove or cause cell death of DNA damage or DNA damage harboring cells in the CNS. We also looked at this in terms of transcriptional disruption. So this is a DNA damage inducing drug known as etoposide. And very much like what we were seeing in ionizing radiation, we also saw that AIM2 and the inflammasome and Gasdermin-D downstream of it were, at least in this in vitro system, were some of the major players in terms of coordinating the cell death in response to DNA damage. Okay, so that's obviously in vitro. We wanted to get a better look at this in an in vivo scenario. So what we did was we looked at P5. So this is a time point when there's lots of DNA damage. And on top of that, we also induce more DNA damage by hitting the P5 mice with pretty high levels of ionizing radiation. But at first, what I'm showing here are aspects. So they're a little hard to see here, but as I showed before in neurodevelopment at P5 in the wild type mice, we see appreciable levels of speck formation. However, when you get rid of AIM2, this is diminished. You can see that here. When you do hit these mice with high levels of ionizing radiation, you can see that this causes an increase in speck formation, as you would expect, due to the DNA damage that's being produced. And then if you get rid of the AIM2 sensor, so this DNA damage sensor, you can see that that leads to greatly reduced levels of speck formation in response to DNA damage. In terms of cell death, obviously, as I told you before, during development, there's lots of cell death that's occurring, and this cell death is actually required to form a properly developed and functioning brain. And so you can see, looking at tunnel as a marker of various different forms of cell death, you can see in the wild type brain, this is actually in the cerebellum, that there are innumerable aspects found in the wild type mice. But when you get rid of AIM2, you lose many of these. It's not all, it's probably not the major way that cells are undergoing dieback, but it seems like a certain fraction of the cells that undergo dieback during neurodevelopment are being driven down that execution pathway by DNA damage and the AIM2 inflammasome. Okay, so this is under basal or homeostatic settings or situations. When we do hit them pretty hard with an induction of DNA damage using gamma radiation, you can see that, as expected, this drives a lot of cell death in the developing brain. So there's lots of tunnel staining. And much of this is lost, not all of it, when you get rid of this DNA damage sensor, suggesting that in response to overt DNA damage, AIM2 is driving inflammasome activation, that speck formation, and also the purging of at least some of the DNA damage harboring cells, although it's unlikely to be all of them. Okay, so this is good. This shows basically what I said and what we were looking at, but obviously there are caveats to the in vitro system and then this very high level of DNA damage induction. So we next wanted to evaluate it in terms of a steady state conditions. Were inflammasomes being activated in response to DNA damage? And then what are the consequences of this? So we went back to our favorite time point, this P5, so 5 days after birth, a point where we know that there's a lot of dieback occurring and that there's lots of DNA damage. And this is looking at DNA damage and speck formation in the cerebellum in the Ask reporter mice. And we have various different markers for DNA damage. So we have gamma H2AX and also 53BP1. And you can see, as I mentioned before, the developing brain is basically littered with DNA damage. And interestingly, when you kind of zoom in on the aspects that are present in the developing brain, many of them are in very close proximity to cells that stain both for gamma H2AX and 53BP1. So these two markers of DNA damage, suggesting that there is a possibility that AIM2 could be, or the inflammasome could be engaged in response to DNA damage that develops in the developing brain. So much like what I showed before, so this is looking at tunnel staining, cell death, and this is at P5. You can see when you get rid of AIM2 that this reduces the total levels of tunnel staining. It's not complete. We think that this inflammasome kind of fail-safe death pathway is kind of a backup in case apoptosis, for whatever reason, can't get the job done. And what we're still kind of figuring out, we think that obviously apoptosis is a major pathway that causes dieback of cells in the developing brain, but AIM2 can also play a role in certain scenarios, and we're still trying to figure out what the exact scenarios are. So ICE, so caspase-1, 11 double knockout, and also this executioner of inflammasomes cell death known as Gasdermin-D. we also see, reduced or statistically significant reductions in cell death when you get rid of these. And what happens as a consequence of getting rid of AIM2, caspase 1 and 11, and then Gasdermin-D is that in certain neuron populations, in certain areas of brain, we actually see that there's more neurons in these mice that lack these AIM2 inflammasome players. So this is looking at Purkinje cells, which are in the cerebellum. And you can see that there's, in mice that lack this pathway, they have much higher levels of these specialized calbindin positive neuronal cells known as Purkinje cells. So if the theory is correct that the AIM2 inflammasome is helping the developing brain to get rid of DNA damage harboring cells, we would also expect to see that in the absence of this pathway, that there would be higher levels of DNA damage. And that is what we see. So this is comparing a wild type to an AIM2 mouse. And this is the gamma H2AX DNA damage staining that I showed you earlier, or marker that I introduced earlier. And we can see that in the absence of AIM2, there's really high levels of DNA damage in various places throughout the brain. We've also validated this by other methods. So this is using a common assay, and you can see that there's much more of the DNA damage in the tail, which is indicative of DNA damage. And then we also see more DNA damage in mice that lack caspase 1 and 11. So I'm gonna take a drink real quick. Okay, so everything I've shown you up to this point has been done in a germline full body knockout. So the possibility exists that some of the phenotypes that we're seeing on the brain are due to deficiencies in the periphery. And one scenario you can think of was if you don't have AIM2 or inflammasomes in the gut, you could have dysbiosis, so changes in your microbiome, and various groups, including Marco Prince and Sarkis Mazmanian, who I think presented in this series a long time ago. But basically, the microbiome composition during development can also shape brain maturation and some of the things we're seeing. So we wanted to see if this pathway was being engaged intrinsically in the brain, or if it was kind of a consequence of not having AIM2 in the periphery. So the way that we did this was we got the caspase 1 flox mice who were graciously supplied to us from the Flavel lab, and we crossed them onto a nestin Cree. So nestin will hit basically all brain cells except microglia. So this was our way to kind of address if we do knock out caspase 1 in the brain specifically, was this sufficient to generate some of the behaviors, increased DNA damage and alterations in cell death that we're seeing. And what we found when you compare caspase 1 flox mice that are Cree negative to those that express Cree behind the nestin promoter, that the mice that lack caspase 1 in the brain in all cells except microglia, they have higher levels of DNA damage as shown by the gamma H2AX stain. These mice that have conditional deletion of caspase 1 in CNS cells also show the anxiety-related behaviors. So this is the elevated plus maze, where mice that are anxious or thought to be anxious don't go out into the open arms. They're also showed this anxiety-related behavior in the open field. So these caspase 1 deficient mice, the conditionals spend less time in the center, and they also have poor motor coordination. So our working model at this point is that during neurodevelopment, there's DNA damage induction. Many of the cells undergo likely apoptosis, but some of the cells that don't undergo apoptosis as quickly likely activate the AIM2 inflammasome. And this provides a way to purge the brain of these cells that have DNA damage, and contributes to the development of a healthy brain, and the incorporation of these genomically compromised cells in the brain. So when you don't have AIM2, caspase-1, or Gasdermin-D, this can lead to DNA damage accumulation, impaired removal of these genomically compromised cells. And then the ultimate result of this is that there's altered brain maturation, and then this could contribute to behavior abnormalities. So I wanted to thank the lab. This is one of the most current pictures of our group. This was at a really great symposium that was held at WashU a couple months ago. It sounds like they're going to have it again in a couple of years. I would highly recommend it. It was a blast. But Kat's actually not in this picture, but Kat did all the work. Wanted to highlight Richard Flavelle, who gave us the caspase-1 conditionals, Veit, who gave us AIM2 conditionals, which I didn't show. Big thanks, as always, to Yoni Kipnis, who kind of took an immunologist under his wing and kind of taught him how to do behavior and to think more like a neuroscientist. He's still been a really great help to our lab and a lot of others. And then Scott helped us with the DNA damage common assay. Tony Filiano, who's now at Duke and is doing great. He helped us with a lot of behavior tests. And then Anton Laveau, who recently started his lab at the Cleveland Clinic, he was very instrumental in getting us off the ground in terms of neuroimaging and various other things. So big shout out to him too. So with that, I'll take any questions. I can, if there are questions.
Yeah, this is great. Thank you, John,
Yeah.
for the wonderful talk.